Hi, I'm Ellie Dimambro-Denson and I'm the Monitoring Officer for Cairngorms Connect. And this evening I'm going to be talking about the two rare plant invigoration projects we are currently completing, which focus on Montane willows, mainly downy willow, and Twinflower. But first, a bit of background to help introduce the project. So Cairngorms Connect is a habitat restoration project covering 600 square kilometres of the Cairngorms National Park. And to help introduce some of the habitats and visions of the project, I'm going to play a short video. So Cairngorms Connect is a large-scale habitat restoration project happening across a continuous connected area of 600 square kilometres in the Cairngorms National Park, making it the largest habitat restoration project happening in Britain. And it's been funded for its first 5 years through the Endangered Landscapes programme. We're currently now about 3 years into the project. The project is a partnership taking in 4 partners: the RSPB, covering its Abernethy and Inchmarshes reserves, which are the areas in pale blue on this map here on the left. Forestry Inland Scotland, or FLS, which are the areas in green. Nature Scot, which are the areas in pink, and Wildland Limited, which are the areas in dark blue. Each of the partners are making their own decisions, but working collaboratively towards shared objectives and a shared 200-year vision, working together for the landscape and for nature. The partnership covers a vast array of different habitats within its 600 square kilometres, ranging from vast tracts of ancient Caledonian pine woods, wild rivers, near natural floodplains down at altitudes of 200 metres, and onwards up the wooded slopes, moorland and near natural tree lines, up onto the most extensive subalpine plateau in Britain, and all the way up to 1,309 metres above sea level to the summit of Ben Macdui, the second highest mountain in Britain. So the project encompasses an extraordinarily diverse and spectacular landscape and a huge array of habitats and species that call it home. So the broad restoration goals for Cairngorms Connect are to expand the woodland to its natural limit and restore the ecological processes within. So this includes restructuring plantation origin Scots pine woodland to create deadwood largely absent historically, removing non-native species, and investing in both natural regeneration and in adding missing tree species into the forest expansion zone and around the high alpine scrub to establish new woodland. Over the 200-year span of the project, this could see an increase from 13,000 hectares to 26,300 hectares of woodland. The project also aims to restore 1,400 hectares of peatland and 900 hectares of bog woodland to capture carbon. Peatlands are amazing carbon stores, storing about a third of the world's overall carbon, which is more than all the forests in the world combined. But because of how they've been managed, many have become seriously eroded and exposed to air, releasing carbon far faster than it can be locked away. So through restoring peatland, this helps to capture carbon and prevent it from being released. And the project also aims to restore 1,000 hectares of floodplains through removing embankments and water control structures over 514 hectares. And finally, through increasing public awareness of the importance of the area and enhancing the livelihoods, as well as the well-being of local people, such as yourselves. And so there's multiple elements to the project, which are summarised here in this spider diagram, each of which needs monitoring to ensure that we can learn the most from the project. So one aspect is through specific monitoring projects to help track the progress of the restoration journey, whether that's through using species such as moths or deadwood beetles as indicators to track their responses to habitat change, or monitoring water flow and quality as the peatlands are restored, and also the public's perception around restoration over time. As a part of the work, we've also been completing a number of management trials to looking to restore the ecological function of woodland through creating deadwood or seed sources of infrequent broadleaf species and improving the field layer for capercaillie productivity. Within this, there are two rare plant species within the project that were identified as requiring intervention to help ensure their long-term survival as part of a rare plant invigoration project. So these two species are Montane willows, more broadly, but more specifically downy willow or Salix laponum, which is seen in this photo here on the left, which has largely become restricted to a small number of isolated and inaccessible cliffside patches, and so become vulnerable to becoming lost. And our second species is Twinflower, which is similarly fragmented in isolated populations that are scattered along the forest floor beyond the distance of pollinators to ensure successful cross-fertilisation and seed production. So first, an overview of the genetic rescue work that we've carried out so far on Twinflower, focusing more specifically on the Twinflower reintroduction project that we completed in 2020. So some background. Twinflower is a small creeping dwarf plant that grows by sending out long runners, which are also known as dolans, over the ground. It's found in boreal forests in the northern hemisphere, as well as mountainous forested areas at more southerly latitudes. It predominantly grows in Scots pine forests, but can also be found amongst birch and on open heathland too. It's smaller than macro photos often capture it to be, with the flowering stalks usually only being about 10 centimetres or so tall. In the UK, it's a nationally scarce species and listed as both a BAP and RSPB priority species, and as well as featuring on the Scottish Biodiversity List. It's predominantly distributed in the northeast of Scotland, with the Cairngorms comprising its main stronghold. So why is it so scarce within the UK? Well, a long history of habitat loss and fragmentation within the Scottish landscape has caused Twinflower to become confined to small relic sites that are highly isolated from neighbouring sites. Although it can reproduce asexually and so persist, pollinators are unable to travel the distances between neighbouring sites, which means that there's very little seed production and therefore new genetically distinct seedlings that are able to disperse. This then means that individual patches of twinflower tend to become genetically isolated. In fact, a study from 2016 found that 80% of patches tend to just comprise of a single genetic individual, with the other 20% usually containing only closely related individuals. This makes patches very vulnerable to extinction in the long term through environmental change or disease. Without seed production, it's also unable to disperse to new areas of suitable habitat, and so the relic populations are unlikely ever to cross without intervention. Therefore, by translocating twinflower, this allows isolated populations to come into contact again, produce seed, and thereby restore genetic diversity of twinflower and allowing it to both recover and spread into the long-term future. So there's been active twinflower conservation work happening within the Cairngorms since 2010, and in 2014, the Cairngorms Rare Plant Project, led by the then project officer Andy Scobie at Nature Scott, completed a series of twinflower translocations in Abernethy using two different translocation methods. So the first method was through reintroductions of twinflower, where two neighbouring blocks containing twinflower clones, which are runners that contain identical genetic material, from 6 different sources were planted into new areas of suitable habitat within pollinating distance of its neighbours, such as in this diagram here on the left. These were also known as the phase 1 trials. The second method was through reinforcements, where additional clones were planted around the outside of a pre-existing natural patch of twinflower that was isolated, again with the new plots within pollination distance to again increase the chances of cross-pollination and seed production between the natural patch and its new neighbours. These were also known as the phase 2 trials. In the summer of 2019, we completed the 5-year post-translocation monitoring for the patches in the phase 1 and phase 2 trials. These included looking at the density of twinflower within the plot, the maximum area or spread of the twinflower beyond the plot, which was a 2 by 2 metre square, the flowering stems that were present, and if any of those flowering stalks had managed to produce any seed. We also took some broader measures of the plot, such as the surrounding vegetation types and their heights as well. We found that although the plots had taken some time to do so, almost all had successfully established, and there was a promising level of flowering and even some seed production within the translocated plots. Following on from this success, we decided to add some additional clones to 3 of those phase 2 twinflower patches in the autumn of 2019, so that each patch had a total of 8 additional clones surrounding it to further increase the genetic diversity of the sites. So that was following the method that's seen here on the right in the reinforcement phase 2 trials. So rather than having just 5 squares around the outside, we then had a total of 8 planting plots around the outside. Then following on from this work, we wanted to complete a trial on a larger scale and decided to focus on the reintroduction method to establish new populations within the Cairngorms Connect area. So here's a closer look at the design for the twinflower reintroduction blocks. Essentially, it comprises of 6 plots, each within 10 metres of its neighbouring plot, which is seen to be the maximum optimum distance for pollinators to be able to successfully pollinate between patches. The plots are arranged along a north-south, east-west grid for easier identification of individual plots and also for design consistency between blocks. Each of the plots is comprised of a 2 by 2 metre square with the corners marked using wooden marker posts and containing 16 individual transplants, which were individual stolons, all from the same clone in the same donor patch, which were planted at each of the 16 marked planting locations, which you can see in the right. So in the diagram on the left, each of those different coloured squares are all a different clone, and within each of the plots, that's all from the same donor patch of Twinflower. So the first stage of the project was to select some suitable sites for the reintroductions. Twinflower tends to prefer areas with a very short, predominantly mossy field layer and a moderate density of canopy cover, usually about 60 to 80%, that's ideally herb-rich with moderate moisture levels. At each site, we were looking for suitable habitat across 2 areas, at least 0.1 hectares in size, to form a pair of blocks containing 6 translocation plots, which were both within 50 to 200 metres of one another. We chose 4 sites in Abernethy and 1 on Glenmore to set up the 5 pairs or 10 blocks. Andy Scobie then helped us to identify 20 suitable Twinflower patches that could be used as donors within 20 kilometres of the recipient sites, which are the red dots on the map here on the right. As a lot of the natural patches on Abernethy had been used in the previous translocations, this meant collecting some donors from further afield than in the previous trials to avoid over-collection from the natural patches, or also by reducing the genetic variation at these new sites by planting closely related genetic individuals together. Where possible, the donor patches also came from at least 1.5 kilometres apart, as studies have indicated that within that distance, individuals tend to be related at the first cousin level, which isn't ideal for maximising genetic diversity. So we use stolons for direct translocations as opposed to collecting and propagating material to later plant out, as the 2 methods are generally equally successful, but direct translocations here reduce the biosecurity risk and also the time aspect involved. In the direct translocations, individual runners were gently uprooted from the ground and picked. Stolons selected for up to about 40 centimetres long, with at least 1 set of well-developed roots and ideally some branching to the main stem, as in this example here on the left. As every donor patch was used at 3 recipient sites, 48 individuals were collected at each of the 20 collection sites, so that a total of 16 individuals could be planted in each of these recipient plots. The Twinflower was then stored in Ziploc bags containing soggy cotton wool balls to stop them from drying out overnight while they were out of the ground, and they were stored in a shed at home overnight before being planted at the recipient plot within 3 days of being uprooted. To plant the stolons, we dug some shallow trenches about 3 to 4 centimetres deep and ensured that the roots were covered and the stolons secured in place amongst the surrounding vegetation. And essentially we repeated this to plant all 16 individuals within a single plot. And so here is an example of a plot after planting. At each block, there's 6 plots that were planted in as close to the configuration as the diagram in the top left as a field layer allowed. Through the autumn, we planted 60 plots such as the one above, and a total of 960 individual stolons. As the stolons planted are relatively small and partially buried, they're not the easiest to spot right away here, especially if you're looking at this on a small screen and you're looking amongst the vegetation, but they can just about be made out at each of these white planting markers. And in 5 years' time, we might expect it to look a bit more like this as the stolons continue to grow and spread, increasing in density and cover within each of the plots, and eventually cross-pollinate to be able to produce seed. So these photos are both from some of the phase 1 reintroduction sites completed in 2014, when we were completing their 5-year monitoring 18 months ago. The photo on the left shows a big increase in vegetative propagation from the single stolons planted, and on the right you can see where some seed has managed to set. So what is next for Twinflower? Well, the next steps here will be to monitor the translocations. We've now completed the first round of monitoring for the 2019 translocations during the summer of 2021, and we're currently processing the data. And the first round of monitoring for the reintroduction translocations will be carried out this summer in 2022, after they've had a full growing season in their new homes. And with other ongoing projects targeting twinflower conservation, such as Plant Life's recent lottery funding for the Rare Plant Wild Connections project and work by NTS at Mar Lodge, it is looking like a really hopeful time for twinflower within the Cairngorms. So our second species that we've targeted within our Rare Plant Invigoration project is downy willow, or Salix laponum, a red book dated vulnerable species, which is one of 7 Montane specialist willow species that we're targeting within this project more broadly as a group. It's found in isolated patches as part of the Montane scrub zone and can occur from about 450 to 1,100 metres above sea level in Scotland, depending on the climatic conditions. And it can grow to about 1 metre tall, and the leaves are downy below, leading to its name. So Montane woodland and scrub forms the transition zone between the forest timberline, which is the upper extent at which trees can be grown commercially, and the heath, or exposed rock, where conditions become too exposed for the shrubs to be able to survive. In natural landscapes, this is often a gradual feathering as different species go through their altitudinal limits. But land management in Scotland often means that this is seen more as a harsh edge, and it's often actually missing. So at lower altitudes in the zone, you can often see the twisted crumholts forms of trees such as Scots pine or downy birch. But as you travel up to the higher altitudes in the zone, it's generally comprised of specialist montane species such as dwarf birch and montane willows. Although again, these species are often absent due to historic land management. So Montane willow scrub is one of the rarest habitats in the UK, restricted to just a handful of small isolated fragments with restricted distribution within Scotland. It's an important and underrepresented habitat, providing food and shelter for specialist plants, invertebrates and other animals in exposed conditions. Of the Montane willow species, both downy willow, or Salix laponum, and woolly willow, or Salix lanatra, are listed as vulnerable, and wattle-leaved willow, or Salix mercenites, is listed as endangered in the Red Data Book. Although we don't know their precise extent within the distant past, the pollen records here have been somewhat inconclusive in determining this. We know that in recent centuries, the coverage of the remaining areas where montane willows can be found has been greatly reduced through changes to climate, grazing and fires, restricting these specialist species to just a handful of sites across Scotland, often in exposed, inaccessible rock ledges out of the reach of grazing animals. Leaving, as with twinflower, isolated patches too far for pollinators to be able to travel between sites. To make things even more difficult for montane willows, they're also all dioecious species, which means that they have male and female flowers which occur on separate trees, and both male and female trees need to be close enough, generally within about 50 metres, which is their sort of optimum pollination distance, for insects to be able to travel between them and allow pollination to occur, and thereby produce the viable seed. Single gender populations may, as with isolated twinflower clones, reproduce vegetatively, but this still keeps them isolated and makes them genetically extremely vulnerable to events in their precarious sites, such as rockfalls, avalanches, disease, or viruses which could wipe out a whole population. Also, as the effects of climate change begin to take effect, montane species are generally going to need to move northwards and to higher altitudes to suitable conditions to ensure their long-term survival. But with limited populations and potential for reproduction, montane willows are unlikely to be robust enough to respond and adapt without conservation intervention. So to better understand the montane willow populations within the Cairngorms, a study by the National Park Authority was funded in 2016 to 17 to survey the existing populations of montane willows in the park. Populations were recorded in Glenfeshie and in the Loch basin of Abernethy within the Cairngorms Connect area. As there was already an existing programme of montane willow conservation work happening on wildland ground in Glenfeshie, the main focus for the Cairngorms Connect genetic rescue work was prioritised to the Loch population on Abernethy. In the basin of Loch 45 willows were recorded across 7 locations. Of these, 37 were Salix laponum or downy willows, which were found across 5 different sites, and the endangered wattle-leaf willow or Salix mercenites had only a single female plant present on the reserve. A genetic study from the Royal Botanic Gardens in Edinburgh found that across the 37 downy willows or Salix laponum, there were 29 genetically distinct individuals, 5 clones and 3 suspected hybrids, showing that there is some remnant genetic diversity, likely a signal that the population was historically part of a much bigger interconnected population. However, all but one of these sites was only comprised of one or very few individuals, and the most diverse population, G, which was found towards the south east of Loch, had 19 individuals, which was actually still contained less than half of what would be needed to be considered a sustainable population, which is about 50 or so genetically distinct individuals, again, all within 50 metres of their nearest neighbours, which is considered to be the maximum successful pollination distance. After these studies, it was decided that a seed orchard would be set up from the cuttings of the mercenites plant, but the short-term focus would be on making the fragments of Salix laponum or downy willow sustainable through augmenting the existing populations in the Loch basin in a similar way to how we did the Twinflower reinforcement project, and setting up new populations in suitable habitat too to facilitate cross-pollination in a similar way to the Twinflower reintroduction project. So in the spring of late 2021, with the help of 30 volunteers from across the partnership, we carried out 3,000 downy willow seedlings, which came from Trees for Life at Dundragon. The 3,000 trees were planted by contractors across 17 plots in areas of suitable habitat around the basin. Some plots which rounded the remnant populations and others in areas of new suitable habitat. The willows were a 50-50 split of males and females and planted into areas of suitable habitat ranging from the crags and ledges where they would be found naturally to richer habitats including flush ground and burnsides in slightly more accessible areas. So on the left here, there's one of the downy willows within its new home in one of the new sites set up. And on the right, if you look really carefully, you'll see 6 willows, each with a temporary marker sort of just above it, planted below one of our natural populations, and Max, who was our research assistant last summer, surveying one to check in on how it's doing. The layout of the plots is more randomised than the Twinflower reintroductions and augmentations to allow for the planters to plant in a range of microsites where they're most likely to be able to survive according to the topography of the site, the soil conditions and the surrounding vegetation. But they generally either followed a linear feature such as a crack line or a gully, or form somewhat of a cluster within suitable habitats. So over the summer, we completed the baseline monitoring for the translocations to accurately record the conditions that they were planted in to help us understand what factors might influence survivability, to help us learn and share this with other projects going into the future. So during the baseline monitoring, we recorded general health, size and location for a subplot, for a sample subplot of the Willows, and carried out vegetation surveys and took soil samples at each of the plots monitored. We also set up camera traps to monitor mammals such as hare and deer that may be browsing the Willows. And here in this photo is Pip, our project scientist, recording the vegetation surrounding one of our sample willows. So this summer, we're planning to head back out again to Loch to plant another 1,500 downy willows. The nature of the site and the decision not to use measures such as protective fences or tree protectors in such a remote area means we're expecting to see a relatively high level of mortality. So it's considered best practice to plant additional willows through the first 3 years to ensure enough survive to be able to form a self-sustaining population. We'll also be completing a more detailed round of monitoring to assess how the willows have fared during their first year within the Loch Basin. And with my lodge working on similar translocations within just a few kilometres of the border, the long-term prospects for downy willow are also looking extremely positive too. And just to say a huge thanks to everyone involved with both projects, and of course to our funders at the Endangered Landscape Programme. And thanks very much to all of you for listening this evening.